So, when Phil invited me, he followed it up with a bunch of very difficult questions, which I didn't have answers for. So what we do normally in academia, we answer questions which are difficult with other questions. And so that's what my talk is going to be about, is to give you more challenges, perhaps at different scales. So I've chosen 3 examples, and these are at the cellular scale, both intracellular as well as the collection of cells. This is inside the workings of a cell where phages are being created and assembled spontaneously. And that's like a closer look at a multi-resolution view of a ribosome. So the brand of visualization that I've been doing, and as well as what motivates the challenges, has not been just to come up with a picture, but also to perhaps support scientific inquiry. So it's always this integrated loop of capturing real data, predominantly from imaging at multiple modalities, modeling that into what we call domain models, and then also building physiological models, and then trying to use simulation to understand the physiological, the form-function relationships. And so, of course, visualization is then used in an interrogative mode, where we can go back and inquire as to how accurate is our results and how accurate and valid is our models. And then, of course, we go into this refinement process. So what you're seeing above is a structural animation of the closed packings within a cell, and I'll tell you more about this, but the yellow-colored things are dendrites, and the whites are axons, and this blue or purplish thing is glial cells. And, of course, nature abhors waste, and you can see how dense the packing is there. What that little clip was of the workings of a ribosome, and the animation that you saw at the bottom was, as this thing continues on, was of a interaction encounter of a molecule which is a potential neutralizing protein that's trying to attach to a static model of a spike protein that's on the envelope of HIV. And the colors are showing electrostatic potential interactions. And what you can see that the binding is occurring, it reduces the overall complementarity of the positive and the negative interactions. So taking a step back then, and in my 10 minutes, I said, well, how do I go back and solve these questions and discover all these form-function relationships? So in each of these scenarios, one goes to what we call back-of-the-envelope calculations first. It said the entire brain, and the Neurophysiome project is to go back and find the entire circuitry of the brain. And after you find the circuitry of the brain, or maybe simultaneously, you try to discover the structure-function relationships. And the brain is huge, even though it's only about 2% of our weight. The entire bunch of neurons are way beyond what we can start to fathom. And so one started to look at then, again, this is with collaborators, and I'll tell you at the very end as to how science is done with collaboration, and that's perhaps my contribution to one of Phil's questions, is Kristen Harris and Dan Johnston at the Center of Learning and Memory, and they've been focused on the hippocampus. And Kristen is the microscopist, and Dan Johnston is the electrophysiologist. And this is ably abetted by Tom Bartol and Terry Cisnowski at Salk. And so they were asking me questions about this 15 years ago, and I had no clues then, but now I do have clues, and I'm coming back to them and asking them other questions. So the fact was that they were focused on a very small region of the hippocampus. They're interested in the entire hippocampus, but they were looking at the CA1 region. And Kristen produced this data set, which is a 6-micron cube sample. It's serial section transmission electron microscopy. The primary reason was also is the cells here, these neurons are, the dendrites are very spiny, and they were trying to understand the role of spines and exactly how does it affect function. So some of the questions we can ask is, how does this local variation in dendritic structure affect the local electrical signals in dendrites? Spines plentiful are frequently absent in disease states, and so where's the critical cutoff where some higher order function suddenly disappears if there's a certain threshold of spines that disappear? So how do we answer these questions? As a computational scientist, you say, oh, well, let's build up a finite element model. And there was one existing, which is MCell, and you'll hear more of that from Terry tomorrow. And he said, okay, you can do a Monte Carlo simulation, but that requires you to build a finite element mesh. And so conservatively, I selected and I said, oh, I need something like 10 to the 10 tets to feed into MCell. And if I was to do even a, what we call linear finite elements, the total memory requirement suddenly turned out to be 1 terabyte. And so in my back of the envelope, the input data was significant, but the computational data that you produce is even larger. And so I needed something like a teraflop machine to go back and do simulations at this scale. And if I was to scale up a little bit larger, which is what we've now received from Kristen, this is using scattering electron micrographs, where it's something like a 50 micron by 50 micron width, then this 60 micron cube data set would require petaflops of communication. So amazingly, I can't solve the problem that I wanted to solve so that I can do this. And so what's the solution? The question was, maybe we have to go into multi-scale models so that we don't solve the problem completely at one scale, but look at the quantities of interest. If you're interested in the spine and the local geometry of this mushroom-like head of the spine, then that's the problem we'll solve at that physics level. And everywhere else, we will derive boundary conditions from a reduced model, which is at a Kirchhoff level model or a cable model scale. So that's one possibility for us to go back in. But it's definitely a challenge. In Texas, we do things big, but sometimes they have a reason for it. And that is, if you start to look at this data, just to discover where synapses occur, there's no geometric rules to tell you where synapses occur. So you have to go back and examine close to these spiny boutons and the axons coming together where post-synaptic density and there's enough vesicles. And that's how you go back and validate some of the models. And that's where we've started to use visualization in an interrogative loop to go back and solve this. The next exercise was something that I had been doing before, together with collaborators, Joachim Frank, Tim Baker at UCSD. You'll start to see UCSD occur quite often, and that's why I'm here quite often as well. And Steve Lutke and Sriram Subramaniam. And this is both single particle cryo-EM, the inverse problem of reconstructing from 2D to 3D, and also cryo-electron tomography, like what Wachu was mentioning, Wachu's student was mentioning earlier. So to just do that interaction encounter of a inhibitor, one has to go back and derive the spike proteins on the envelope through a process of tomographic reconstruction, and then plucking out each one of the spikes and averaging them to come up with an improved model. This entire process of reconstruction itself, if I do back of the envelope calculations, the current state of the art is using something like less than 1,000 squared pixels. And if I want to go down to 1 angstrom resolution, which is what people are trying to do as the holy grail in cryo-EM, can you get atomistic structure? Because currently, the best of models which utilize symmetry are down to 4 angstrom, which is fantastic. It's a great. So again, these calculations show you that we're already testing the limits today, and the questions that we really need to answer is going to make the limits even go further. We perhaps build one of the largest iPads at UT, but it's useful sometimes to interrogate things when you're looking at scales which are right down from the angstrom level to hundreds of thousands of. And then you finally look at the last thing that as my third example, and this is molecular-molecular interactions. And one utilizes dynamics. Again, I'm very thankful for the collaborations with Andy McCammon and Mike Holst, as well as Art Olson and Michelle Sanner. We've known each other for now several years. And so, again, this is well known to to several people in the molecular community, but just doing some simple calculations, saying there's so many different models of molecule-solvent interactions, intramolecular energy, and if you want to really build interrogative models where we can query them and come closer to a good approximations of reality, then this thing is starting to get way out of hand. And given the limitations of time, I won't go into the specifics of Poisson-Boltzmann, but just come up with a rough chart. If I was to solve some of these simulations, dynamics, or even do docking with them, today I can only solve problems in the 1,000 range. And if I wanted to really solve problems up to large assemblies within a cell, in a virtual 3D cell, and support them with these pseudo, even reduced approximation physics, then I'm projecting in 2020 we might be able to solve some of them, but only with reduced models. To do quantum explicit and ab initio for the intramolecular energy and quantum explicit for the molecule solvent, these equations are very hard to solve. But I'm a big fan of human ingenuity, through multi-scale modeling, multi-resolution, multi-physics, and coming up with the right scale bridging. I think there's hope. There's hope because we have to focus on what are the quantities of interest. And it's only for the relevant quantity of interest to define where do we need to go to a finer scale such that we can bootstrap and improve the accuracy. We don't really need to solve everything at one fixed scale. And with that, I'll quit and acknowledge my group, which is interdisciplinary, people from math, computer science, as well as sometimes from engineering as well. But without these collaborations, I don't think I could have succeeded. There's too much to span, and even the little I tried to chew, if it hadn't been for these people, where I can quickly ask a question and get the right answers, or I don't know, this is a good question to answer, is the kind of thing that I think will propel us in the future. So with that, I'll stop, and thank you very much.